Good afternoon, everyone. It's really nice to be here in this beautiful room and really nice to be able to at last present live again. I'm going to focus my talk today on the technology at Spago Nanomedical, the evidence that we have for this technology, and the projects we are running, and provide a status update on those projects. I will also give a flavor of the market that we see, especially in therapeutics. So, at Spago, we have a nanotheranostics platform that is based on physiological targeting of solid tumors. I will not go into details, but basically what we do is that we take advantage of the inherent features of solid tumors when it comes to blood supply and nutrient exchange, which makes it possible for us to tailor functional nanoparticles in a way that they accumulate in tumors and perform what we want them to, whether it's diagnostics or it's therapeutics. So many people have worked with nanoparticles and heard about the concept before. So what's the difference? Well, in our case, I would say functional nanoparticles is what we do and what we can. The particles themselves are enough for us to do what we want at the site of the tumor. And we can use that for diagnostics in the shape of an MRI contrast agent. We can use it for therapeutics in the shape of a radionuclide treatment to actually kill the tumor cells. And I will explain this a little bit more in detail. So, does this work? Well, we have set out to design and produce and test a precision technology that makes it possible to go into tumors very precisely and visualize them or treat them. And we have shown within the framework of the ongoing clinical trial that we have, I will come back to that as well, that our material does exactly that. It does accumulate in the tumors, which makes it possible to visualize it. If you look at the image to the right, you see a breast cancer tumor, very well lightened up by our nanomaterial in an MR machine. It provides a lot of information of the tumor without the background contrast that you normally see with today's contrast agents. So this is really a proof that our technology works and that we can tailor nanoparticles to go to the tumor. So, how do we use this? Well, as I said, we are working with diagnostics in the shape of a contrast agent for MRI. Tumor selective makes it possible to visualize tumors much better than what is possible today. Our second project, and perhaps the most interesting one, is TumorAd, which is a radionuclide treatment where we use the same type of particles to deliver a radiation dose to the tumor inside of the body. The diagnostic project SpagoPix is in clinical trials, and with TumorAd we are now quickly approaching clinical phase. So I will start off by talking a little bit more in detail about SpagoPix and then come into TumorAd. So what's the difference with this contrast agent compared to the gadolinium-based agents that are currently standard on the market? Well, first of all, what we can do is to provide more information about the tumor without showing the background tissue, which today is a problem because what happens is that you will get false diagnosis with the current agents. So with SpagoPix, or SN132D, which is our product candidate, we have shown that we can provide a much clearer image of the tumor. We can clearly show the edges of the tumor. We can even see that if we look at the picture to the right, or the image to the right, there's a dark area in the middle of the tumor, which is likely representing necrosis in the tumor. So what we provide is the potential for increased precision, and in the end, of course, improved patient outcome. And it doesn't stop with breast cancer. We have started in breast cancer since we think this is a good indication, but this is, of course, applicable, as we see it, to other solid tumors as well. What we also have found is that we have a very good contrast in the pancreas and in the liver. If you look at the image, the arrow, the orange arrow indicates the pancreas in this image, and it's a very, very good delineation of the organ that is not possible to do today with the MRI contrast agents that are available. So this is actually an additional opportunity. It provides for high resolution of the pancreas, which is really important if you want to be able to detect early stages of pancreatic cancer. And there is a major clinical need in this area. Of course, as you probably all know, pancreatic cancer is a deadly disease. And there are no MRI agents approved for this indication today. So this is really something that we want to look into further. So where are we? Well, as I said, we have a clinical trial ongoing here in Sweden. It's a phase 1 study in breast cancer patients who are newly diagnosed. It's run in Uppsala and in Gothenburg. As always with early clinical trials, safety is the primary endpoint. But since this is an imaging agent, we have the opportunity to actually collect initial efficacy data in the form of images. So we concluded one dose group, which indicated that we have good safety, and then we proceeded into another higher dose group, which is ongoing, and where we have interim data that shows excellent contrast. Now then, as you all know, the pandemic hit us, and it's been really difficult for us to find patients for the trial, as with many other clinical trials that is ongoing. We hope that that will gradually become better now, and that the situation opens up at the hospitals. And the next analysis will be when we have completed the ongoing dose group. So, but the important thing is here that the interim results that we have already shows that this works. So now switching over to therapy, our second project, Tumorad, and this is when it becomes really interesting, because now we are taking this concept to the next level. We are designing particles that physiologically targets tumors by means of accumulating in the tumor tissue and delivering a radioactive dose to the tumor and killing tumor cells. So this will be a way to irradiate tumors from the inside of the body. And does this work? Well, we have preclinical evidence. We have tested this in an animal model of aggressive breast cancer. We have shown that we can stop tumor growth and extend survival of those animals. And our candidate drug is SN201. That is now progressing towards clinical trials. And I should also say that the particles themselves have very low toxicity. We don't see basically any toxic signs with the particles themselves. The toxicity that is observed will come from the radionuclide in the particles. So that makes it possible to predict safety of these agents. So earlier this year we raised capital with the help of Naventus to fund the acceleration of Tumorad to clinical trials. And that is actually going very well and according to plan. So we are now performing regulatory studies that will build the base for a clinical trial application. We are producing material that will be enough to do a quite extensive clinical program. And we are building the documentation that will be needed for applying for a clinical trial approval. So all of this is happening now in parallel, and the aim is to start clinical trials in the next year. And what it all tells us is that what we promised the market earlier this year was that we're going to bring TumorAd to the market as quickly and as effectively as possible. That's what we are delivering on right now. So, a few words about the market, just to give a flavor, and why am I saying that TumorAd is the more interesting project here? Well, I'm not saying that imaging is not interesting, because it's really, really important to have new contrast agents that can provide better visibility of solid tumors. But when it comes to radiotherapy, there are not so many agents out there that can actually effectively treat tumors inside of the body and provide a new tool that can be added to the toolbox of the doctors when treating cancers that are otherwise difficult to treat. So what we saw a couple of years, or now a number of years ago, was that Spago came along. Spago was really the first example of a new modern radionuclide treatment. Remember that radioisotopes has been used to treat cancer for 50, 60 years. It's nothing new in that sense. Everybody knows that radioactivity works to kill cancer cells. The problem has been to make it accumulate in the tumors in a way that makes it suitable as a therapy. Spago was one of the first examples of a modern radionuclide treatment. It accumulates in bone tissue, which makes it suitable to treat bone metastasis. This was acquired by Bayer in 2014 from the Norwegian company Algeta for $2.9 billion. More recently, and what we are seeing now, is that a few of the large big pharma companies are investing heavily in this segment. And Novartis is the star in this arena. In 2018, they bought two companies and two projects, one in a specialized form of cancer called neuroendocrine tumors, Lutathera. It's now on the market, and sales are increasing year by year. And the second one reported positive phase 3 data this year in prostate cancer, which is expected to reach the market potentially this year or probably next year. So what we are seeing now is that there is this really, if we want to cite the article here in the slide, that these drugs are now coming about and storming the market. A little bit exaggerated perhaps, but at least we are seeing a modern mode of attacking tumors using radioisotopes. So with this, I think we're in a good position. We listed at Nasdaq First North earlier this year. We changed from Spotlight Stock Market to Nasdaq First North, and the reason for that was mainly visibility, and as Niklas explained earlier, to be able to attract new types of investors. And with that, we also made a capitalization which puts us in a good position now to actually execute on what we promised the market. Next out will be the readout of the Spago Pix clinical trial, and then we have a number of milestones coming within the TumorAd project on the way to clinical trials. We will be looking, or we are looking for partners for the Spago Pix projects, because we see the real value in this technology will be, for us, in therapeutics, and that's the way we want to proceed. And eventually, we'd like to grow the pipeline, of course. So I think Spago Nanomedical is well set at this point to become one of the winners that, again, Niklas referred to earlier. We have the technology, we have solid evidence, we have a strong pipeline in both diagnostics and therapy. And we believe that the market speaks for us, especially in therapeutics and in this field. So with that, I'd like to thank you for your attention, and I'm happy to take questions.
Thank you, Mats.
Thank you.
So given the large interest for your company, if you're more specific, what would you like, what are you hoping to achieve in the coming 12 months?
Well, we are, as I was explaining, we are executing on what we promised the market earlier this year. We would like to bring Tumorad to clinical trials as soon as effective as possible. And that's really what we're doing right now. And it's so far, I can tell that it's going really well, and that's proceeding according to plan. That'll be one of our main focuses. The other will be, of course, to finalize the SpagoPix trial, obviously, which we would have done if not the pandemic came about. And obviously sign a partner deal with that project to capitalize on that.
So could we actually, from a patient perspective, in the not so far away future, could a patient first have its tumor diagnosed with the SpagoPix and then be treated with the Tumorad?
That's a very good question, and that's probably true. Although I would say the benefit of having a radioactive treatment is that you can actually use that treatment also as an imaging agent. So having a radioactive treatment that goes to the tumor makes it possible to image it before giving the therapeutic dose. So you can give a smaller dose, see it go to the tumor, and then you say, okay, this lands where we want it to land, and then you go with the therapeutic dose.
So it will be much more specific and direct, the treatment.
So really personalized treatment.
Well, this is very exciting, very promising. Thank you very much.
Thank you very much.